Okay, let's get started. Hello, everyone. On behalf of the SingHealth Duke-NUS Global Health Institute, I'm delighted to welcome all of you to this three-part webinar series. Currently, the world's attention is understandably focused on how to better manage the pandemic. However, in this webinar series, we want to shift the focus to really examine the broader question of why such a virus originated in the first place, and maybe what we can do to prevent further future occurrences. The first webinar, this is a three-part webinar series. The first webinar provided an overview of the role of evolutionary and ecological factors in pandemics like COVID-19. Today, in the second webinar, we look at a One Health approach in the fight against emerging zoonotic infections. We'll explore strategies to prevent emerging infections through controlling the source and explain available tools like surveillance systems. And I hope there's a lot that you guys can learn from this. Our first speaker today is Dr. Ian Mendenhall, who is a faculty with the Emerging Infectious Diseases program at Duke-NUS Medical School and affiliated with our Global Health Institute. Ian is a principal research scientist in the Laboratory of Virus Evolution run by Dr. Gavin Smith. Building on his work on the ecology, evolution, and epidemiology of infectious diseases, he was awarded a grant to develop methods to more efficiently detect bat-borne viruses. Ian has worked in Singapore, Vietnam, Indonesia, Kazakhstan, India, and Cambodia with both government and university labs to build capacity and better inform biosurveillance. He will call on this extensive experience to present the talk today and will also be moderating the session. I will leave it to introduce Dr. Erik Karlsson, who's our second speaker. And just before I do hand over to Ian, I'd like to make a few housekeeping notes. Please note the webinar will be recorded. Keep yourself on mute the entire time. You can pose questions via the chat box at any time during the presentations. At the end of the two presentations, the questions and answers session will start and the questions will be answered at that point. But please feel free to go ahead and post them at any time. So we look forward to kind of having this discussion with Ian and Erik and with all of you. Hand over to you, Ian.
Sure. Well, it's my pleasure and honor to introduce Dr. Erik Karlsson, who is not only a collaborator of mine, but a good friend. So during his doctoral work at the University of North Carolina, he studied the effect of nutrition, especially diet-induced obesity, on immune responses to viral pathogens. Following his PhD, he began to pursue a research career focused on understanding surveillance, virus evolution, host pathogen interactions, and immune responses. His postdoctoral work at St. Jude's Children's Research Hospital in Memphis, Tennessee, focused on virology, viral pathogenesis, co-infection, immunology, and infectious disease surveillance at the animal-human interface. Dr. Karlsson currently serves as a senior researcher at the Institut Pasteur de Cambodia in Phnom Penh and is in charge of all ongoing activities related to respiratory viruses, including human seasonal and zoonotic viruses. He's a director of the National Influenza Center and the regional WHO H5 reference lab, and he helps to manage the WHO COVID-19 Global Reference Laboratory and frequently consults for organizations such as the Food and Agricultural Organization of the United Nations. He's also the founder of the Consortium of Animal Market Networks to assess risk of emerging infectious diseases through enhanced surveillance, otherwise known as CANARIES, and we'll hear a little bit more about that during his talk. So, without further ado, I will get my presentation up and running here. All right, well, thank you everyone for attending. I'm going to talk a bit about wildlife biosurveillance today, and I'm not certain how many people were present last week with Dr. Rudge and Dr. Smith, but I'm going to cover some of the same topics again because I think it's important. So I think we all have a pretty good grasp of One Health, and it's really just looking at human health, animal health, and the health of the environment, and understanding that these are inextricably linked. And you know, it's really important that when we're trying to tackle these larger problems that we're bringing in subject matter experts from multiple fields, because multidisciplinary problems really require multidisciplinary teams. And I've got a great example of that. And this is kind of where I cut my teeth in zoonotic diseases. As I had just finished my undergraduate degree, and that was right at the time when West Nile virus emerged in the United States. And it's the first time that that virus had been there. Now, West Nile virus is a flavivirus, and it just circulates enzootically between mosquitoes and birds, and occasionally it will spill over into incidental or dead-end hosts. So in June of 1999, there was a lot of dead crows. Now, crows are really large birds, and the death of thousands and thousands of these raised some eyes, but no one looked into it very deep. And then a few months later, we saw spillover into incidental hosts, and we had manifestations of encephalitis and acute flaccid paralysis in human and horses in New York City. The problem was that people looking at human health and animal health weren't talking, and that was a large failure and has since been resolved. So again, this is a bit of rehash from what Dr. Rudge presented, but we have to understand that the majority of human infectious disease originated in animals. And we like to call this viral chatter. So we're exposed to animal microbes all the time, bacteria, their fungi, their viruses. Most of the time, we're just not a good fit for these organisms, and they're not able to infect us and reproduce. Oh, apologies for that. So if we look at kind of on the left side of the screen where we have our bat and our rat and our monkey, we have this really diverse wildlife pool of viruses. And there's a lot of host specificity, so there's a lot of viruses that don't have the capacity to infect other species, but we do have some community transmission. Occasionally vectors are involved with fleas, mites, ticks, or mosquitoes. And then when wildlife encounter either humans or domestic species, we see a spillover. And sometimes that's successful, and it's sustained, and sometimes it's not successful. And often then it will spill into humans. So that's why we like to use animal populations as sentinel reservoirs. In terms of cross-species transmissions, as I mentioned before, we have a lot of exposure events, but very few successes. And we can start with these kind of a hierarchy where we look at these ecological barriers. And it's all about we have to share the same space and the same time as an infected natural host. And now natural hosts, there's a few things that are required for that, right? They can't develop a neutralizing antibody response. They can't die quickly because really they need to be available for that virus to spill over. And they can't have contact, and they need to have contact with alternative hosts to have that spillover. And then when we get down to humans or domestic animals actually being infected with these viruses, we have phylogenetic separation where it's sometimes easier for me to be infected with viruses from animals that are more closely related to me. Obviously, some viruses require a particular specificity, but a lot of what it comes down to is the virus surface protein has to be able to interact with the host cell receptor, and that's where we find a lot of either the susceptibility or being refractory to that. And also, once the virus gets inside a cell, it needs to be able to utilize the cell machinery to be able to reproduce. So if we're going to understand zoonosis, we need to understand that these are stochastic processes, and they're really unpredictable because we don't know what's driving or amplifying or dampening transmission. And if you take a look at the graphic on the right-hand side, I think that's a really good illustration of what's going on. And this is with Hendra virus in bats in Australia. And so you can see the purple, that's where the bats live. That's where their range is, but they're not shedding Hendra virus over the entirety of that range. So we need to figure out, okay, where are they shedding that virus? And then once that virus is being shed, how long is it able to survive outside the host? A lot of cross-species transmission from bats ends up being from them chewing on fruit, and then perhaps a pig or a horse will encounter that fruit and become infected, but not all of them will become infected because they need a certain amount of virus to become infected. And so we know very, very little about this. Influenza is probably the best studied zoonotic disease that we know about. And I think it's important that we understand that we can get a sequence data, and that'll kind of build a family tree, and we'll understand the relatedness, but that sequence data is not really going to tell us how that virus is going to behave when it's inside a host. In terms of zoonotic drivers, there's been a lot of work done, a lot of modeling, trying to understand where hotspots are going to be, and South and Southeast Asia seem to be predicted hotspots for the emergence and spillover of emerging infectious diseases. And part of that reason is there's high human density, and there's a lot of land change. And the land change is really important because it's bringing wildlife in contact with humans and livestock and their domestic animals. But also in Southeast Asia, there's a lot of mammal diversity. And with a lot of mammal diversity, you have this co-speciation of viruses and bacteria. And so you have a lot of microbial diversity. We also know that, at least in a predictive models, climate change is going to drive virus sharing because it's going to be changing these habitats. And it's going to be bringing animals in contact who wouldn't have normally come in contact through the land use change and the impact of climate. We also know that in Southeast Asia, we have very large cities in terms of human density that really facilitates transmission. That's really important. And it's not just these large cities. It's also, we've seen an incredible migration from rural areas to these cities. So they're becoming even more dense, but then people are moving between these urban areas and back to their hometown, opportunity for them to bring these pathogens with them. We also need to think about habitat loss and ecological interfaces. Obviously, if we're not interacting with wildlife, the opportunities for zoonotic spillover tend to be quite low. But once we start kind of increasing the number of interfaces, and a lot of this is due through logging or through agriculture, now we're kind of exposing ourselves to that wildlife virus diverse pool. But we're also stressing these animals out. Because now there's not as many resources from them. So in addition to increasing exposure, we also have potentially stressed species, which may result in increased shedding. So in terms of biosurveillance approaches, I thought it would be good just to kind of group these, lump these into three kind of major categories. And one of them I would consider longitudinal surveillance. And that's really taking a specific site or a specific species and monitoring it over time. So I can go to like a bat roost and I can visit it every 2 weeks. I can visit it every month to look at, is there increased shedding? Is there a change in the composition of bats? Is there a change in the viral diversity there? You know, and that's going to give me an idea, you know, are there temporal trends? And if I can see a correlation with perhaps, does it associate with animal pregnancy periods? Does it associate with drought? Does it associate with resource abundance or paucity? And then I can start to say, all right, these are areas where we may have higher risks of shedding. And those are times where we may want to go proactively survey to see what's going on. The second one I call opportunistic surveillance, and it's really kind of giving you a snapshot because you're going out to a specific place at a specific time and understanding what's going on. And so I can go to a bat cave and I can collect, you know, 300, 400 bats over a couple of nights. I can see what the viruses are there, but it's really just a snapshot. The other part of this is going to kind of high-risk interface areas. So these would be wildlife markets or trade areas where you can go and see, has there been spillover? And obviously, these are places that can also be monitored longitudinally. The third is kind of probabilistic surveillance, and we're looking at kind of spatial temporal trends, and this is what I'm interested in. I'm really interested in, can we build tools to inform biosurveillance? It's not cheap to go out and catch animals and look for viruses and understand the risk of spillover. But if we can kind of give tools where we should inform us to allocate our resources, that's what I'm chasing. So I thought I'd just kind of give you a brief rundown of some of the challenges that we have. And I've been catching animals and looking for viruses and exposure since 1999, mostly in North and South America. And then when I moved here in 2010, I've been working in Singapore on mosquitoes and small mammals and bats. But when you're in the field, you obviously have to deal with these environmental variables. Often it can be quite hot. We're wearing PPE. We need to take care. You can have thunderstorms moving in. Another concern is just the expense in field visits. Often in Singapore, it's quite easy where you can go visit a field site, you can bring your samples back and have them in the -80, and then you can go sleep in your own bed, which is quite a comfort. But often when I'm working in neighboring countries, I don't have that luxury. And also there's a lot of permitting acquisition that really needs to happen. You have to make sure that you have your permission to sample animals, to collect information from humans, and also to be able to visit the different sites. Now, we're banking all of this on the hope that animals are going to show up, and they're going to show up in our traps. And I've definitely had a number of nights where I haven't caught anything, and that's really frustrating. But you know, sometimes zeros are as good as ones, because really we want to understand who's there and who's not there. Now, when you catch an animal, you're handling it, it's stressed, it's frightened, it's going to try and bite you, and so that's a risk. After we've collected the samples, we need to ensure that we've got cold chain integrity, so when they get back to the lab, we're working with kind of viable specimens. And when we're in the field, we're working with sharps and chemicals. And one of the biggest questions that we have out there is how can I tell an animal's sick? At least with some livestock, you have a good idea that they're sick, but with wildlife, you're often not catching sick animals because those are the ones that are going to sequester themselves, so they're not going to be predated. But I think that, you know, we're starting to be able to build some tools that are going to give us a finer scale of resolution about if an animal is stressed by looking at immunological markers that are a bit less coarse than corticosteroids or ectoparasite infestation. Now, once I get my samples back to the lab, I have to think, okay, what are they stored in and what biosafety level do I need to work on? If I'm catching, you know, Pteropus bats, and I know that Nipah virus is endemic in those populations, I'm gonna have to take a lot more care, both in the collection in the field and in how I'm gonna handle them in the lab. And I also have to decide how I'm gonna test, and I know that there was a question that came up, and I'll address this later in the Q&A part, but I have to figure out, am I just gonna be looking for all coronaviruses, in which I'll use a family-specific conventional PCR, or do I have a very specific coronavirus that I know circulates in that population and I can detect, and I wanna kind of quantify? Now, I can do some exploration through next generation sequencing or trying to generate a full genome, or I can try and isolate this virus and grow it up so I have a stock, or I can just look for exposure. Look for the fact that perhaps this animal has been exposed to that virus, and so that's something that we're interested in. Now I've kind of covered the nitty-gritty. I just want to give you a couple examples of a couple projects that I am in and help develop. And obviously, when I say I, it's more the royal I, because we've got a really large team in the Laboratory of Evolution at Duke-NUS, and this type of work does not happen with just individuals. So the first project I'd like to talk about is some of my work in India on a particular village, and they have an annual tradition of harvesting bats. And that's a real kind of fantastic interface because we know that there's going to be high risk exposure. Now, this particular location is located in the northeast of India in Nagaland, and our particular field site, it's about 15 kilometers from the Myanmar border. So when we land, it's a good 20-hour plus drive on switchbacks up and down mountains, and it's a pretty arduous place to get to, but there's a real history there. You know, we have over 150 years and 7 generations of bat harvesting. And for me, it's the most amazing cohort. We have the same 2 bat species, and we have the same individuals 1 day a year going to this cave to collect bats for consumption. And it's fascinating. So primarily, we're looking for evidence of spillover. We're using serology and PCR, and we're interested in filoviruses, which are your Ebola virus and your Marburg viruses, coronaviruses, and your paramyxoviruses. Now what they do is people are only allowed to visit this cave 1 day a year, and it's one clan that owns the cave, and they go in and they light fires and they asphyxiate these bats, and as they're trying to escape, they kill them. And then they round up and they count the bats and they give them out to all the male members of this clan. Because we're working with a PhD student from the National Center for Biological Sciences in Bangalore, who is Naga, it really facilitated being able to contact these folks and be able to work there, but also to be able to provide some education on some of the risks of exposure to bat-borne viruses. And so once these bats are killed, we take some blood samples, and then we put them in a cool box, and we move them all down to our processing field site, and then we take a collection of different organs from each individual bat. What we've also been able to do is take surveys from the humans and also collect sera from the humans. And so we're interested, has there been cross-species exposure? Now this slide is a little busy. And so at the top is human serology, so Homo sapiens. And if you look at the x-axis, what we have is we have surface proteins from all of the different filoviruses. So we have Ebola virus, Bunyibudya virus, Typhorus, Sudan, Marburg virus, and other ones. And then if you look at the little gray dots, those are individual human serum samples. And so what we're interested in is, has that human been exposed to an Ebola virus or a Marburg virus or a related virus? And if it has, they should have generated antibodies that are going to be reactive against those glycoproteins. So if we look at figure A at the top, we can see that there are at least 3 humans who have been exposed to some type of filovirus that's most closely related to something in the genus Ebola virus. Now, because there seems to be a lot of diversity in the filovirus family in South and Southeast Asia, and we know that Ebola virus species doesn't exist there, they've likely been exposed to some undescribed filovirus. Now, if we move down into the panel B, that's Eonycterus spalea, that's the cave nectar bat, and we can see one individual has the same pattern of cross-reactivity exposure to these filovirus surface proteins as the humans. Also interesting, if we move over to panel C, we can look at this other fruit bat, Rosetta schleschnaltii, and it looks like there's two individuals that have been exposed to two different filoviruses. So it looks like there's 3 different filoviruses circulating in these bat populations, and one of them, and we have serological evidence to support this, has spilled over from bats to humans. And so these high-risk interfaces can give you real interesting data, but it doesn't seem like there's been any outbreak, any hemorrhagic symptoms there. So we're trying to understand, we're still working on this, what these viruses are, and if they aren't very pathogenic, why aren't they pathogenic? I'm going to move on to the second project that I mentioned, and then I'll hand it over to Erik. And so this is a project that Gavin Smith is the principal investigator on, and I'm a project manager, but we have a team of 20 plus that are working on this. And really, the goal of this project is we want to develop countrywide maps in Cambodia to study the presence and absence of bats and rats, their ectoparasites, and then a select set of zoonotic viruses and bacteria. Because we really, again, we don't want to sample all over the country or just throw out a fishing net. We want to have specific hotspots that we can visit that should have known higher probabilities of occurrence for the reservoir and the parasites of interest. And we're also studying spillover in humans. So you can see the map of Cambodia. We work with a quantitative ecologist, and he built a model that randomly selects sites that's stratified against land cover types. So we're just not picking sites on a map. These are random sites, and it is a pain to get out to a number of them. So our field team goes out there, they collect bats at night, they collect rats in the morning, they process them all, and then they move to the next site. So it's a very data-hungry model. We're starting to build some really, really interesting results. And so we do have some lab work. I'm not going to talk about that because we're still analyzing it, but I'll get to some of our field research later. But it's been a major, major project. We've sampled in almost every province. We've had 3 years of sampling. We visited 220 sites that the model has chosen, but we've also visited 21 opportunistic sites. And so these would include kind of, rat traders or bat guano harvesters, because those are kind of high risk interfaces for spillover. Sampled over 3,000 animals, a number of catch and release, a number of voucher specimens. We've caught all the small mammal species in the country. We've caught nearly every bat species in the country, and we have a number of new country records, which are quite interesting. And we've got a freezer full of almost 34,000 samples. So we're screening these, and this is going to be able to be built on for a number of years to interrogate. We also have a lot of human sera, and so we're just kind of in the depth of trying to analyze and understand what we have here. In addition to catching bats and rats with traps, we're also taking acoustic recordings, because some bats are really difficult to catch. They go right up into the canopy and you're never going to get them, but we want to know if they occupy the site. And so with this animal collection data, we're able to build these maps. And so on the left-hand side, we've got Rattus tanazumi, which is a really common rat, and Mus cervicolor, which is a mouse. And on the right side, we've got two different genera of bats. And so if the color is red, there's a very high probability that that species exists at a particular site in Cambodia. And if it's blue, there's a very low probability. So what we're doing is we're trying to build these species occurrence maps. And then what we're going to do is build vertebrate community occurrence maps. And on top of that, we're going to layer ectoparasites and viruses and bacteria. So at some point, we're going to have kind of a hotspot map that will tell you if you're interested in surveying for tick-borne bacterial parasites in small mammals, these are the places you should focus your efforts on in Cambodia. And we're hoping to bring this out regionally, and hopefully we'll start working in Laos next year using these same models. So just a few takeaways. We know that EIDs, zoonotic diseases, are very complex. You really have to think about how you're going to design your study before you go in there, and spend a lot of time talking to people. Before we started this project, I sent hundreds of emails out trying to pick people's brains so we wouldn't encounter the same issues that they had to before. And again, failure to incorporate new information will come back to haunt you. So I wanted to thank everyone for that, and I'm going to stop sharing and hand it over to Erik.
All right, there we go. So thanks, Dr. Mendenhall, for that excellent presentation. Luckily, Ian gave all the background information, so I can just talk about some birds and things like that in Cambodia. Again, my name is Erik Karlsson. I am Senior Research at the Virology Unit in Institut Pasteur in Cambodia, and I am very interested in the animal-human interface, especially when it comes to the domestic animal-human interface. And we'll talk a little bit about why. Sorry, there we go. So I really wanted to start with this point right here, which in my opinion has to do with some of the things we just talked about. So when we're talking about spillover, we're talking about a lot of these diseases lately. We're talking about these diseases basically going from an individual wild animal or a bird into a human, into a domestic animal, and causing an individual outbreak. But in my opinion, what also happens is also these endemic periods where we get a spillover event, and then that pathogen then circulates in a secondary host for long periods of time, and then increases the risk for humans. So I always, as Ian mentioned, my background is actually in nutrition. I always like to start sort of talking about food. And I don't know who has been to their mom's house lately or to their grandparents or anything like that, but usually the first thing that you hear in this region is, have you eaten yet? And literally, if you map this out to every single country in Asia, the Western Pacific and Southeast Asia, pretty much one of the greetings is, have you eaten, or have you eaten rice? And we're really not sure why this has occurred, but it's probably because of all the famines and food shortages in the past, but also because food is such an important part of the culture in this region, and sharing food and growing food and sustaining life. So if we look at the global increases in basically domestic animal production, especially in this region, we see that from 1960 until about now, they've greatly increased, especially in developing countries like in Southeast Asia. So if you look in developing countries alone, between 1965 and 2015 about, chicken production's actually increased 567%. Goat production has increased 188%. Cattle production has increased 49%. And the list goes on and on and on. And this is really because these developing countries are becoming more developed, and the demand for protein has become higher and higher and higher and higher. However, what that means is that there's more hosts for things to spill over into and become endemic. So you'll probably remember this image from Ian's talk, where we're talking about spillover. So now we're in this green box right here. So we're talking about viruses that spill over into domestic animals that then become established in domestic animals, circulate over and over and over again, and then have a chance to jump into humans. We also have to remember, say, for coronaviruses, since we are mentioning COVID in these talks, is that all of the human seasonal coronaviruses, so those 4 up at the top right, actually went through an intermediate host, and a lot of those are domestic animals. Even SARS and MERS went through technically domestic animals. So civets, this SARS went through civet cats, which were being farmed, and then MERS obviously went through camels, which are domestic animals in the Middle East. SADS, we're not quite sure if it caused human infections, but that was also an infection in pigs. So what we really know is that domestic animals can act as intermediate hosts for endemic pathogens that can spill over into humans to cause outbreaks and further pandemics. And so I think my numbers are slightly different than Ian's possibly, but 60 to 75% of emerging infectious diseases are found in humans, and they're zoonotic, that are found in humans are zoonotic. And there's a number of reasons why these things happen. Ian gave a very, very nice overview of things like vectors and bats, monkeys. But here, we're talking about domestic animals. And so here in Southeast Asia, there are majority of the production of farmed animals, a lot of them on backyard farms. So this is 2012 data. I probably should update it, but as I understand, as of 2012 in Southeast Asia, 43% of the world's chickens were farmed here, 60% of the world's pigs, and 9 out of every 10 ducks were grown in the region. And so there's a number of other factors that contribute to possible emergence of disease from domestic livestock into humans because of interaction with wildlife. So first is diseases of poverty. We have crowding, we have overpopulation, we have health issues, there's access to healthcare, general biosafety, biosecurity, urbanization, and even air travel. And I think Dr. Rudge and Dr. Smith talked about that last time. So in terms of what we're going to talk about today, I'm really going to focus on avian influenza because that's been a major problem in the region since 1997. It became obviously endemic in the region in 2004, 2005. So we'll focus really on these live bird markets and how avian influenza has spread throughout them with a focus on Cambodia. So there's a number of risk factors that we get for domestic animal spillover, both for diseases to spill over into domestic animals, but also for them to spill into humans after that. So the first is species density. I just gave you some very high numbers about how many animals are actually being farmed here in the region. In addition, at these live animal markets, so this is Takeo Live Animal Market in Phnom Penh, you see that they're very closely packed together. There's not much separation between the animals as well. In addition, we have species mixing. So we have a lot of different species that are housed under the same roof. A lot of these backyard farms in the region are housing chickens, ducks, cattle, pigs, maybe a turkey or two, some geese, fish, and a number of dogs, cats, and a number of other animals in a low biosafety, biosecurity environment. So they have a lot of interaction both with wild animals as well as with each other, as well as with the humans that take care of them. So that brings me to something called proximity. So again, there's a lot of issues with people handling these animals and then handling other animals. So humans as a species can actually transfer these pathogens back and forth to animals, even though they may not get infected. So you have densely packed animals who are kept in groups of a number of different species that are then in close proximity to humans. You also have issues with biosafety and biosecurity. So this is a very common slaughterhouse in the back of a live bird market in Phnom Penh. You see here, no one's wearing gloves, no one's wearing face masks, they're probably not cleaning as well as they probably should. And there's a lot of things going on here that generates aerosols, that generates fomites and things like that, that potentially can create pathogen reservoirs and then spread into humans that are here in the market. Sorry, guys. And then third, the last thing that I really wanted to bring into this is really the human component. So as Dr. Mendenhall mentioned, the risk of a pathogen jumping into a human really depends on a lot of factors, including whether or not that pathogen can actually replicate in a human and how close they are to the animal. But it also depends on our society. So things like malnutrition, things like obesity, things like smoking, things like traveling from place to place. Those can all be factors in the exposure and the risk of a human actually contracting disease, as well as the virus or pathogen and the exposure to that pathogen. So just to move on to a little bit of data, we'll talk about some avian influenza in Cambodia. Obviously, Cambodia is located in Southeast Asia, in case you didn't know. The capital city is Phnom Penh, and the population in the entire country is about 16 million. About 79% of the population lives in rural areas, and in 2015, about 57% of all Cambodian households had agricultural holdings, and 87% of those raised poultry, many on backyard farms with really, really minimal biosafety and biosecurity measures. We also know that poultry diseases can have really devastating economic consequences, which can then lead to a vicious cycle of poverty and infectious disease. So as you get more infections, more outbreaks, That makes people, the economic loss is greater. The people then can't buy the food that they need, and that they become more susceptible to infectious diseases, and on and on and on. So up top, you see poultry outbreaks in Cambodia between 2000, avian influenza outbreaks. This is in Cambodia between 2004 and 2020, and then human cases, which we've had 56 of between 2005 and 2014. The majority of them were between 2013 and 2014, when we had a clade switch between 112 and 2321C and had a reassortant virus. And our case fatality rate is about 66%. So because of this endemicity in Cambodia since 2004, we have been doing live bird market surveillance in a One Health way. So these would be the longitudinal and the opportunistic studies that Ian was talking about since 2006, but really in this longitudinal opportunistic way since 2011. And most of these have been focused in the southern part of the country. You see the red dots down at the bottom, where there's a lot of poultry movement between Vietnam and Cambodia, and where the majority of people live. So our main goals for this surveillance really are to understand seasonal circulation and prevalence, to subtype what's in the country, and then to do things like human seroprevalence to look for spillover, and then also to do things like market supply chain tracking, because that's part of that human component that I was talking about. You can't just, when you're talking about a domestic agricultural system, you can't just look in the market. You have to look at where these birds are coming from, how they're getting to the market, how they're transported, the cold chain, where they might stop over in the middle, and I'll talk a little bit more about that. So here I've just mapped out all of the data we have between 2013 and half of 2019. This data represents about probably about 20 samples from 20,000 birds or more. And you can see there's not really a point in Cambodia when we have zero prevalence of avian influenza in any of these birds. You do end up seeing distinct patterns. So you can see this nice peak in February. There's some minor peaks around August, September. It goes up a little in December as well. And really, what we're trying to do now is correlate that to factors that we can then plug into our models that generate risk. So first thing that we wanted to look at was seasonality. So we have very distinct dry and rainy seasons here. We're in rainy season now. It was raining earlier, actually. And what you see is that that big peak comes in dry season around February, but there are still peaks in rainy seasons. That doesn't really exactly correlate with what's going on. But interestingly, when we overlay something like religious festivals or festivals in general, so here we have Lunar New Year, also called Chinese New Year. We have Khmer New Year, Chumben, which is also called Hungry Ghost Festival, things like that, and then Bonum Tuk, which is Water Festival. You start to see a pattern emerge where you see these increases in avian influenza prevalence in the markets around these festivals. And so what we're researching now is really how that works. And what we think is that because so many people are getting together with family, they're making food offerings at the temples, at the grave sites, they are having parties. We're seeing a lot more increase in poultry production during these times, a lot more increase in poultry movement, and that really helps to spread the avian influenza around the country. So the next thing I really just wanted to touch on, because we don't have a lot of time, and I'm happy to answer questions via email or to talk on the phone at any point in time if people are interested, but was to look at the poultry value chain in Cambodia. So this is a very typical middleman here in Cambodia. I call it the truck of death. But basically these birds have been gavaged with a rice and water solution to make them heavier so that they are actually worth more when they're sold. And then they're hung upside down over these other birds on this truck. What you don't see here is that they're now regurgitating this water on these other birds and spreading the virus around everything else. And so you see these kinds of trucks, you see motorcycles with birds hanging off of them, and then you see these large commercial industrial trucks driving around all the time. And this isn't just for birds. We see trucks full of rats, we see trucks full of cattle, trucks full of pigs. So the value chains here in Cambodia really deserve some attention in terms of zoonotic risk. So what we were able to do is actually convince some of these middlemen to let us LoJack their vehicles. Basically, we put real-time GPS trackers on them and followed them around for about 12 weeks, mainly around the two main festival periods, Lunar New Year and Khmer New Year. And so we were able to get several different kinds of middlemen involved in this. One was local middlemen, which collects the live poultry from backyard farms in the province. They bring it to the provincial live bird market, and then they'll usually buy some there, take it back to their house. There's cross-market middlemen who are collecting from either backyard farms or the provincial live bird markets and then bringing it to storage places or to the main market in Phnom Penh or in Takeo. And then there's cross-border middlemen who are picking up chickens and ducks from Vietnam and Thailand, mainly from large companies, but also from some small traders and things like that, packing them all into big trucks, then bringing them down to provincial markets or storage areas, and then those are spread out again throughout the country. And so you end up seeing these very distinct patterns, and these are not roadway maps just because I didn't want to implicate anything, but this is at the village level. So each dot on this represents a village or a stop on these middlemen's journeys. And you can see that they can get quite complex. So if we zoom in a little bit further, this is just the sort of provincial middlemen, the smaller middlemen. And so this is 3 different middlemen. And what you see is that the red individual doesn't go so far, and the brown and the green individuals go a little bit farther. The entire catchment area here is about 30 kilometers. And you can see how many stops they're making. And this is cleaned up data. So we actually had to go through and make sure each of the stops that we found was either a farm or a storage facility, which was quite an endeavor. The black squares here that you see on your screen are actually the house of the middlemen. The houses of these middlemen become these small live bird market type situations themselves, where they're actually bringing poultry in the evening to their house. They're slaughtering half of them to bring as poultry products. They're taking the other half back to the main market, which is the triangle right here. And then in the evening, if they don't sell what they maybe brought to the market, they'll bring them back to their house. They'll go get some more birds from other markets. They'll bring them there. And you can see how these cycles continue. What's very interesting is that we actually were able to find 2 meeting points that we did not know before. And these actually ended up being novel poultry storage facilities where they're actually bringing poultry to store for 1 to 2 days at a time. And you actually see that multiple middlemen are actually visiting these storage areas. And we didn't know that they were there before. So these actually help us to generate new spots where we have to do surveillance to understand how these viruses are spreading around the country. And then finally, I wanted to talk about human spillover in regards to live bird markets. I don't really have any data here, but we have been longitudinally following all of these slaughterers, these sellers, these middlemen since 2017. So we've actually been able to get serum from them longitudinally 4 times a year since 2017, and that continues to be ongoing. We have not seen too much spillover yet, but it does seem that these individuals, compared to community individuals, are actually exposed to avian influenza viruses at a higher frequency, which is not unexpected. So then I also wanted to give you a reason why we do this kind of surveillance and monitor these things. So to talk about H7. So I know we've all heard about probably subtype A, H7, and 9. We've never detected H7 and 9 in Cambodia. However, H7 and 9 or H7 viruses have spread all over the world and caused intermittent zoonotic transfer to mammals. They cause highly pathogenic outbreaks in birds, and they're highly mutable. So why we're talking about this one now, excuse me, in December of 2017, a 68-year-old woman in Jiangsu province in China entered an ICU with severe pneumonia. She was diagnosed with avian influenza virus subtype A, H7N4. This had nothing to do with H7N9. It was a completely different clade. First case worldwide. Similar viruses were detected in poultry that she had close contact with, and aquatic poultry in Jiangsu. And again, the whole genome analysis indicated it was a distinct lineage. What was very interesting is that we were doing surveillance at the time. And so this is the case in Jiangsu. And so we first detected this virus in Cambodia about 2 months following that case. So it quickly spread across Southeast Asia, and we had not detected this subtype up until this point. So somehow it quickly spread to Cambodia, and now we are still detecting it in our live bird markets intermittently throughout the year. And it continues to evolve. So this is a high-risk pathogen that we're quite worried about. So just to sum everything up, and I apologize that I'm going a little long, where do we go from here? So how do we use these as a One Health framework? We know pathogens continue to circulate in wild domestic animals. We really need vigilant surveillance in a One Health context, not just in animals, but also in humans. Sorry, guys. We really need to share information in a timely fashion. We need to continually reevaluate risk. And it takes a village. As Dr. Mendenhall says, this takes a lot of people. It takes a lot of coordination. It takes a lot of talking. But if we can have these networks, what we can do is actually link them together, okay? So I have this network in Cambodia. Ian has this network in Singapore and other places. And we can network them together. And that's why we generated this consortium, which really focuses on future pandemics and being able to assess risk of emerging infectious diseases by enhancing surveillance by joining together. And I've just included the website down below in case anyone's interested in it. And with that, I'll say thank you. And I'll hand it back to Ian.
Well, thanks, Eric, for a fascinating talk. And I think we're open to questions now. All right, so I've got a question. Have you been able to sequence or isolate any filoviruses from that bat population in India? And the answer is unfortunately no, as of yet. We're still working on generating sequence data. We're using family-specific PCRs. We actually just got kind of an interesting enhancement set, so we're hoping to employ that there so we can, if these viruses are at low levels in the bats, be able to pull them out and see what they are.
Ian, I think there's a question right now from Gavin. If you just look at the chat box and then.
Sure. So Gavin's question is, and there's a couple before that which we'll address, is how can information on exposure risk be used to reduce or remove risk of transmission to humans? So I think when we're looking at wildlife parasites like viruses and bacteria, it's really difficult to know if they have the capacity to infect humans, and oftentimes these can be difficult to grow in the lab into an isolate and do animal challenges. I think if Eric's still online, I think that his experience with AI, he would have some good thoughts on this.
I'm sorry, Ian, one more time.
So this would be Gavin's question, is how can information on exposure risk to be used to reduce or remove risk of transmission to humans? I think education would definitely be one.
So we, in our hands, what we use that risk generation to do is to then target interventions that we can test. So whether that be educational opportunities for, say, live bird market workers to increase hand washing and things like that, to build new markets where they're cleaner, to increase market cleaning. But we also know that if we end up with a cluster of virus or a cluster of cases in that area, that we have to be very careful and be able to monitor those people very closely, because they are at highest risk for those zoonotic spillovers.
I'll answer a question Vibol asked. Hi, Vibol. I can't wait to get back to Phnom Penh so we can catch up. Thanks for your help in the collaboration. He's asking, with the hotspot maps that we're generating, is there going to be an online web link available? And obviously we're not in the infancy, but we're in the middle of generating these, and the goal is to make an interactive map. So you're actually going to be able to click on the provinces and understand what animals we caught there, what viruses we found there, what they look like genetically, and then we want that to be a tool, obviously, for anyone to use, and that's the goal. And then Fahad has a question on how would you respond to the rumors that viruses are being deliberately spread with political motivation? I mean, this is a challenging question, because I think there's, in today's kind of mass media and easy access to information, it's the signal to noise ratio, right? So there's so much noise going on, and how do you vet it? And right, so you can just point them to scientific papers, but they may not be digestible. And I think it comes down to parsimony, kind of what's the most reasonable explanation that you would see this. And I think if you look at SARS coronavirus 2, you know, we can look at the genetics of the virus, and we can say that the most likely explanation is this was not some lab generated infection. But it's a really difficult question. I don't know, Eric, do you have any thoughts on that? On how would you respond to rumors that viruses are being deliberately spread?
I agree that there's a lot of signal to noise here. I think that a lot of these things happen often, and people are looking for explanations for them. And we have to understand that the natural processes really do occur, and especially since we're having increased populations, increased animal populations, climate change, things like that, that are pushing us further and further towards interacting with the potential areas of spillover, that these things are inevitable to happen naturally. I'll also answer the question from Debbie right there real quick, because then I'm going to have to go to give another seminar. But they ask, how is the data being generated by our groups get used by health authorities to support response or preparedness? I know from our avian influenza studies and things like that, that goes directly into things like understanding what vaccines are being generated, or providing data for the global information system for influenza, as well as other databases that can then be used to understand risk. So they're directly used by a lot of animal facilities, a lot of groups, to understand the drift of influenza viruses and how we can change our vaccines, or what new vaccines may be needed in certain areas, or even what people might be at the best choice for things like clinical studies for avian influenza vaccines, or even for animal vaccines. So with that, thank you, everyone. I actually have to go, but I will talk to you later.
Thanks, Eric.
Thanks so much, Eric. Great to have you on.
Sure, I'll hang around and I'll answer some more questions. So we had one actually before the presentation that came in a couple days ago, and it was, would doing surveillance on bats for coronaviruses be sufficient, especially since many coronaviruses require intermediate hosts to jump to humans? And so usually when people are surveying animal populations for coronaviruses, you're using a detection method that's focusing on a very conserved part of the virus. And so that's how you're able to capture kind of a lot of genetic diversity in a lot of different hosts. The problem is when you start to see changes in the virus genome that allow it to spill over into other animals, that happens in the spike protein. And that's very divergent across coronaviruses. And there's not, unless you have that virus sequence of the entire genome, it's really difficult to be able to understand if this virus may have the capacity. But what I think just doing a general survey for coronaviruses with a conventional PCR gives you is it gives you enough information to say, oh, look, this coronavirus from this bat or rat species, it sits in a group of other coronaviruses that we know have been implicated in cross-species transmission before. So it may be one that I want to interrogate further. And then just one more quick thing on the how does the data get used by health authorities. In the majority of the projects that I work on, it's overseas. We try and work with government agencies so that that information is going right into the stakeholders' hands and they're able to use it. At least in Cambodia with the maps we're going to generate, we want those to go to all of the affected partners who are working on One Health and emerging infectious diseases. Brian has a question on, have you seen any impacts of climate change in your longitudinal studies? If so, can you describe what you think the important variables might be? We haven't in any of our studies. Usually when you're looking at kind of space and time, it's really expensive and difficult to incorporate both of those. So in Cambodia, we're primarily looking at a spatial aspect. In terms of temporal, there's a PhD student in our lab, Dulcie Lowe, and we did a lot of surveillance of myna birds in Singapore and looking for a particular virus shedding. And we were looking at, is there an impact on rainfall? Is there an impact, in their breeding season? And we haven't seen that. I think that those variables are difficult to suss out unless you have like really long-term studies, and those are often difficult to fund. I'm sorry I don't have a more satisfying answer for you. Erik has
Ian, I'll actually jump in on that and just kind of remind everyone that the third part of our webinar series actually, not in a nitty-gritty way, but kind of at the broad level does look at some of the ways that maybe we can interact, internationally, globally, and link these very large questions of environment, climate change, environmental impact, ways of production, food systems, so we will have a webinar that will address that at a broad level. It won't go into the details, but we do hope that people will get some insight and have some hope of being able to prevent these occurrences. So just remind everyone to log in for that.
All right, I'll move on down the line. So Erik has a question: can all these research and resources be able to lead us to predict future COVID-19 outbreaks? And I don't think they're going to allow us to predict them, but what we're going to get is they're going to be building kind of these evidence banks. So if you look at what happened after the first SARS outbreak is people understood that bats are a very rich reservoir of coronaviruses, and a lot of folks started doing surveillance in bats. And by doing so, they started to discover a lot of coronavirus diversity. And the group at Wuhan Institute of Virology, they've been doing longitudinal studies on specific caves. And by doing so, they've been able to characterize kind of the genetic diversity of coronaviruses, and they have kind of what appears to be, the ancestral bat coronavirus. So I think that once we understand kind of who the intermediate host might be, who might be susceptible, we can start to focus our resources on those areas. But again, it's really challenging because I think if anyone thought there was going to be another pandemic, it would likely be influenza. So it's quite interesting. Eleanor has a question: based on your surveillance, have you identified key or immediate interventions that would make the most impact to reduce the spread of disease? That's a really good question, but it's really challenging because I think at some point you have to take each virus kind of in a silo because they're transmitted by different hosts that have different behaviors, and we have different interfaces with them. And again, as I mentioned before during my talk, is the majority of these animal viruses, we're just not a good fit or a home for them. They're never going to be able to infect us. But at least looking at, bat viruses, we know that filoviruses, coronaviruses, paramyxoviruses, and reoviruses, those are groups that have shown that they can spill over to us. So we can focus on species of interest, and we can look for those particular viruses. Narayana, and please, I apologize if I pronounce anyone else's name wrong. Apart from hand hygiene and social distancing, will being vegan, vegetarian prevent the occurrence of these wildlife and animal acquired diseases? I think certainly for animals that are spilling over into wildlife, when they end up in wildlife markets, obviously these animals are stressed and they're encountering other animals that they're not going to naturally encounter in nature. So that's something I think that, and I'm not a vegetarian or a vegan myself, but I think that the intensive animal production facilities certainly lead to opportunities for great overspillover from wild ducks, who are the natural reservoir for all avian influenzas, into these kind of domestic duck and domestic chickens. So I think kind of understanding, if not the entire world can become vegetarian, but you know, if we can reduce our reliance on meat, but that's something that's quite difficult to say, because if you're kind of someone who's a subsistence farmer, obviously we need to be able to ensure that they're able to feed their families. I think that may have been the last question. Does anyone else have any other questions? Otherwise, my email is online. I'm quite happy to respond to emails, and
yeah. So we'll just close with a thank you to Ian and to Erik in absentia for this very interesting kind of talk, which shows the great complexity of trying to figure out the interplay between the animal host and human spillover effects. And we hope that everyone will sign up for our webinar 3, which, as I said, would give you some kind of broad outlines of how to prevent future pandemics, hopefully. So thank you, everyone. Thanks to the speakers, to the admin team, and to all the participants. Bye-bye.